produced in factories. This is why they are sometimes called designer antibodies. They are tailor-made to the disease they treat. The first monoclonal antibodies produced was licensed more than 30 years ago. Since then, millions of people have benefited from more than 100 such treatments. Around 50 of these were brought to market in the past 6 years alone. This is one of the fastest growing fields in biomedical research and increasingly important segment of the pharmaceutical market. Last year, 7 of the top best-selling drugs were monoclonal antibodies. How do they work? Antibodies are protein produced by our immune system and are one of the main ways the body defends itself against diseases. They work by binding to their specific targets, for example, virus, bacteria, or cancerous cell, and making them harmless. They block the action of the target or they flag it as foreign so that other parts of our immune system can clear the invaders away. Monoclonal antibodies work in the same way too. They bind to the specific target without harming anything else in their way. This target is not always a foreign intruder like a virus. Antibodies can be designed to attach to different molecules in the body, for example, to tone down the immune response, which is with when it overreacts. This phenomenon, which also happens with some COVID-19 patients, is called cytokine storm. Due to their numerous applications, monoclonal antibodies have been safely and effectively used to treat a growing number of diseases, some of which were difficult to treat in the past. Monoclonal antibodies work by binding to their specific targets, for example, viruses, bacteria, or cancerous cells, and making them harmless. Monoclonal antibodies bind to their target. Once attached, they make it harmless. How are they made? Monoclonal antibodies is complex and expensive. First, scientists extract the relevant antibodies from human blood, then they replicate and manufacture them in large quantities. Most monoclonal antibodies are produced in Chinese hamster ovary cells, which are typically grown in large bioreactors for around 10 to 15 days. The resulting antibodies are then purified and packaged so they can be easily administered. The whole process takes a long time and uses costly materials. Some estimate the cost of producing 1 gram of marketed monoclonal antibodies is between $95 and $200. This doesn't include the cost of research and development or packaging, delivering, and administrating the medicine. The costs are even higher for startup companies. Manufacturers are looking into ways to reduce production costs, for example, through novel technologies and using alternatives to hamster cells such as algae, yeast, and plant. They would change the way these drugs are made. COVID-19 monoclonal antibodies for treatment of COVID-19. For the past 30 years, monoclonal antibodies have transformed the way we treat various diseases. They prove to be more effective, better tolerated, and easier to deliver than other treatments. Researchers are optimistic that monoclonal antibodies could help prevent and treat early infection of COVID-19 too. Some of the advantages they offer are they can specifically target the SARS-CoV-2 virus because they originate in the blood of people who have recovered from COVID-19. This would potentially make them more effective than other drugs. They can be rapidly isolated and manufactured. Since the pandemic started, more than 70 monoclonal antibody products for COVID-19 are now in development. They can provide rapid protection against infection. Once administered, monoclonal antibodies enter the bloodstream straight away and offer immediate protection for a few weeks or months. Vaccines they take a few weeks to have an effect but usually provide long-term protection. This is why researchers are optimistic that monoclonal antibodies could complement vaccine in helping to contain the pandemic. They have the potential to treat infected patients or prevent infection in all individuals, including the elderly and young children and immunocompromised people, some of whom can't receive a vaccine or for whom vaccines don't always work as well. How many monoclonal antibodies for COVID-19 are in development? Since the start of pandemic, researchers have been rapidly evaluating existing drugs and developing new treatments, including monoclonal antibodies to treat COVID-19 patients. Several monoclonal antibodies that are licensed or in development for other diseases are in clinical trials to see if they have an effect on COVID-19 patients. One of these is adalimumab used to treat arthritis or Crohn's disease. The University of Oxford recently launched a trial to look at its potential to treat people in care homes funded by COVID-19 Therapeutics Accelerator. Researchers have also been rapidly identifying monoclonal antibodies that specifically target SARS-CoV-2. More than 70 such products are already in development. The pharmaceutical company Lilly, in collaboration with AbCellera, launched the first human study of a potential COVID-19 antibody treatment in May-June this year. Other safety clinical trials followed, including studies of AstraZeneca, Celltrion, and Regeneron. Thank you.
खै त एनी क्वेसन भनेर सोध न साथीहरूलाई के
Questions?
क्वेसन छ भने सोध खै केही सोध्नु छैन साथीलाई रुबीलाई केही सोध्नु छैन
म एउटा क्वेसन सोध्छु है त यसमा चाहिँ होइन अब टपिक नै मोनोक्लोनल एन्टिबडिज फर द ट्रिटमेन्ट अफ कोभिड-19 भनेपछि चाहिँ कुन कुन एन्टिबडी बनाएको रहेछ त कोभिड-19 बनाउँदा यहाँनिर चाहिँ मेन्सन गरेर चाहिँ डिफ्रेन्ट टाइप्स अफ के अरे चाहिँ प्रडक्ट्सहरू हेज बिन ट्रायल जुन चाहिँ मोनोक्लोनल एन्टिबडिज हो कोभिड-19को ट्रिटमेन्टको लागि तर कुन कुन हो भनेर मेन्सनै गरेको छैन can you give me some names of monoclonal antibodies? हेलो म्याम मेरो आवाज आइरहेछ
मैले सुनिरहेको छु रुबी खै त रुबीले एन्सर थाहा छ कि थाहा छैन रुबी
थाहा छैन म्याम
अरू कसैलाई थाहा छ त डिस्कसन पनि हुनु पर्यो है, डिस्कसन पनि हुनु पर्यो। उनलाई थाहा छैन भने अरू कसैलाई थाहा छ भने यस्तो यस्तो छ है भनेर भन्न पनि पर्छ है। मैले अस्ति उसमै लेखिदिएको छु, हाम्रो के अरे स्लाइडमै छ त्यो मोनोक्लोनल एन्टिबडिजको एउटा नाम, के अरे ट्यान्जिनोमाब भन्ने समथिङ के छ, हेर त, त्यो एउटा मलाई थाहा भएको। अरू मैले त्यो नाम लेख्दै थिएँ होइन र स्लाइडमा एउटा, मैले त्यो स्लाइड बिर्सेँ।
त्यो म्याम हाइब्रिडोमा टेक्नोलोजी पढ्दाखेरि होला।
हो, त्यो एउटा चाहिँ त्यो नामको मोनोक्लोनल एन्टिबडी भयो होइन। अरू त्यस्तै अब तिमीलाई त्यस्तै नाम चाहिएको होइन?
हजुर म्याम।
धेरै एन्टिबडीहरू छ, जस्तै रिप्रो भन्ने छ, हिमिरा छ है, सिमुलेट भन्ने छ, पेन्सेल्टा भन्ने छ, पाल्भा छ। त्यो साउन्ड सुनिरहेको छ?
एकदम छैन।
ल ठिकै छ, अब उसको प्रेजेन्टेसन सक्यो है। अब नाम चाहिँ त्यही यसमा हेर है, नेटमै पाइन्छ होला होइन, मोनोक्लोनल एन्टिबडिज फर कोभिड-19 भनेर पाइन्छ नेटमा पनि होइन। अब एउटा चाहिँ त्यो अस्ति मैले त्यही लेखिदिएको त्यो एउटा भइहाल्यो, अनि अरू चाहिँ त्यही नेटमा सर्च गरौँ होइन, त्यही पाइन्छ होला। अरू अब नेक्स्ट प्रेजेन्टेसन चाहिँ हाम्रो कसको थियो अरे, शारदाको है। शारदा
हजुर म्याम।
रेडी छ होइन?
हजुर।
हुन्छ, ल स्टार्ट गर्दा हुन्छ। शारदाले प्रेजेन्ट गर्न लाग्यो हो
म्याम हो तर मेरो स्लाइडै सेयर भइरहेको छैन
ल्यापटपबाट हो
हजुर
यस्तो गरौँ त त्यहाँबाट मिलेन भने मलाई मेल पठाइहाल अनि म यताबाट देखाइदिन्छु
हुन्छ म्याम
है ल शारदा मलाई पठायो
म्याम म पठाउँदैछु म्याम आयो?
यस आयो है, म खोल्दैछु।
स्टार्ट गरौँ? नेक्स्ट स्लाइड।
ल गर्दा हुन्छ है। स्लाइड आयो हैन?
हजुर आयो।
ल
Good morning everyone. I am going to present some knowledge what I know about the useful product obtained from cell lines. Ma'am, next slide. Cell lines are often used in place of primary cells to study biological process. Different भ्याक्सिन्सहरूको प्रोडक्सनको लागि ड्रग्स मेटाबोलिजम्सको अनि साइटोटक्सिसिटी अनि एन्टिबडी प्रोडक्सनको लागि सेल लाइन्सहरू युज गरिन्छ। म्याम नेक्स्ट स्लाइड। सेल लाइन्सहरू तीनटा टाइप्सको हुन्छ। फाइनाइट सेल लाइन्स जसलाई नर्मल सेल लाइन्स पनि भनिन्छ, कन्टिन्युअस सेल लाइन्स अनि ट्रान्सफर्म सेल लाइन्स। फाइनाइट सेल लाइन्स चाहिँ 5 to 10 times डिभाइड हुन्छ। Normally, cell usually divide only a limited number of time before losing their ability to proliferate, which is a genetically determined event non-sense. फाइनाइट सेल लाइन्सको इक्जाम्पल भनेको मङ्की किडनी सेल लाइन्स हो अनि चिकन इम्ब्रियो सेल लाइन हो। अनि कन्टिन्युअस सेल लाइन चाहिँ कन्टिन्युअस सेल लाइनको इक्जाम्पल भनेको क्यान्सरस सेल लाइन्स हो, इमोर्टल सेल लाइन्सहरू हो। Continuous cell lines, when a finite cell lines undergo transformations and acquire the ability to divide indefinite. Continuous cell lines लाई indefinite cell lines पनि भन्छ। अनि transformed cell line, conversions of a normal health cells into tumorous and cancerous cells. अनि transformed cell lines लाई stem cell lines पनि भन्छ। Ma'am, next slide. Cell culture. Cell culture refers to the removal of cell from an animal or plant and their subsequent growth in a favorable artificial environment. Cell culture गरेर सेल लाइन्सबाट सेल कल्चर गरेर डिफ्रेन्ट भ्याक्सिन्सहरू अनि पेप्टाइड्सहरू पनि बनाउने गरिन्छ। फर डिफ्रेन्ट यसको पर्पज डिफ्रेन्स मेथडहरू युज गरेर यसबाट भ्याक्सिन्सहरू नि निकालिन्छ, ड्रग्सहरू पनि निकाल्ने गरिन्छ। द सेल्स मे बी रिमुभ फ्रम द टिस्यू डाइरेक्टली एन्ड disaggregated by enzymatic or mechanical means before cultivation, or they may be divided from a cell lines or cell stain that have already been established. Ma'am, next slide. From animal cellsबाट मोनोक्लोनल एन्टिबोडिज हाइब्रिडोमा टेक्नोलोजीबाट मोनोक्लोनल एन्टिबोडीहरू पनि बनाउने गरिन्छ। अनि इम्युनो बायोलोजिकल रेगुलेसनबाट इन्टरफेरेन एन्टी क्यान्सरस अनि ग्लाइकोप्रोटिन्सहरू पनि सेक्रेट गर्ने गरिन्छ। अनि भाइरल भ्याक्सिनहरू पनि निकाल्ने गरिन्छ अनि हर्मोन्सहरू निकाल्ने गरिन्छ फ्रम एनिमल सेल्स। मोनोक्लोनल एन्टिबोडी अन्तर्गत मेनली हाइब्रिडोमा टेक्नोलोजीबाट मोनोक्लोनल एन्टिबोडी निकालिन्छ। अनि त्यो चाहिँ डायग्नोस्टिक्स एसेस सिस्टम डिटर्मिनेसन्स ड्रग्स टक्सिन्स एन्ड भिटामिन्सहरू। मोनोक्लोनल एन्टिबोडीबाट मेनली निकालिएको एन्टिबोडिज, थेरापेटिक्स प्रोसेसबाट निकालेको एन्टिबोडिज भनेको एन्टिलिपोपोलिस्याकाराइड हो, जुन चाहिँ ट्रिटमेन्ट अफ सेप्सिसको ट्रिटमेन्टको लागि युज गरिन्छ। सेप्सिस भनेको बोडी एक्सट्रिम रेस्पोन्स टु एन इन्फेक्सन भनेर बुझ्ने गरिन्छ। अनि अर्को भनेको बी सेल्स लिम्फोमा भन्ने छ। बी सेल्स लिम्फोमाको बी सेल्स लिम्फोमा भन्ने अर्को एन्टिबोडी हो, मोनोक्लोनल एन्टिबोडी, जुन चाहिँ बी सेल लिम्फोमाको एप्लिकेसन यसलाई युज गर्ने गरिन्छ। अनि डायग्नोसिस चाहिँ गरेको भनेको ब्लड क्यान्सरको लागि नि डायग्नोसिस गरेको छ भ्याक्सिन्सहरू निकालिएको छ मोनोक्लोनल एन्टीबाट। अनि त्यो भनेको 99 TEMFAB फर ब्रेस्ट क्यान्सरको लागि पनि निकालिएको छ। अनि हर्मोन्सहरू भनेको हर्मोन के अरे एनिमल सेलबाट निकालेको हर्मोन भनेको इराइथ्रोप्रोटिन्स हो। इराइथ्रोप्रोटिन्स दुई टाइप्सको छ, इराइथ्रोप्रोटिन्स ए भन्ने छ, इराइथ्रोप्रोटिन्स पी भन्ने छ, जुन चाहिँ एनिमियामा युज गर्ने गरिन्छ। अनि ह्युमन ग्रोथ हर्मोन्सहरू पनि प्रोड्युस भएको छ। त्यसपछि अरू भनेको भ्याक्सिनहरू भनेको HIV भ्याक्सिन्सहरू, मलेरियल भ्याक्सिन अनि पोलियो भ्याक्सिन्सहरू पनि एनिमल सेलबाट नै निकालिएको छ। म्याम नेक्स्ट स्लाइड। एनिमल सेल अन्तर्गत चाहिँ म्यामेलियन सेल र नन म्यामेलियन सेल छन्। म्यामेलियन सेल्स भनेको एनिमल आइसोलेटेड फ्रम म्यामेलियन एनिमल्स हो, जसलाई चाहिँ कल्चर गरेर रिसर्च टुल्सको रूपमा प्रयोग गरिन्छ फर प्रोडक्सन्स अफ भाइरस भ्याक्सिन भेरियस थेरापेटिक प्रोटिन्सहरूको लागि। म्यामेलियन सेल्स अन्तर्गत पनि ह्युमरल सेल्स र नन ह्युमरल सेल्स भन्ने हुन्छ। ह्युमरल सेल भनेको ह्युमनको सेल लाइन्सहरूबाट निकालिएको ह्युमरल सेल्सहरू हो भने नन ह्युमरल भनेको म्यामेलियन सेल तर ह्युमन बाहेक अरू एनिमल्सहरूबाट निकालिएकोलाई चाहिँ नन ह्युमरल सेल्स भनिन्छ। त्यसको सेल लाइन्स र प्रोडक्ट्सहरू भनेको ह्युमन ट्युमरको लागि एनागोनिक फ्याक्टर्स अनि ह्युमन लिम्फोसाइटको लागि इन्टरफेरोन्सहरू छ। म्याम नेक्स्ट स्लाइड। म्यामेलियन सेल लाइन्स चाहिँ होस्ट सेलको रूपमा म्यानुफ्याक्चर हुन्छ जब हामीले धेरै बायोथेरापेटिक्स प्रोटिन्सहरू निकालिन्छ। तर म्यामेलियन ले अरू होस्टहरू पनि प्रिफर गर्छ, जस्तै ब्याक्टेरिया, प्लान्ट्स एन्ड इस्ट, बिकज अफ देयर क्यापेबिलिटी फर प्रोपर प्रोटिन फोल्डिङ एसेन्सली एन्ड पोस्ट ट्रान्सलेसन मोडिफिकेसनको लागि। अनि यहाँ दिएको इक्जाम्पलहरू भनेको चाहिँ के अरे म्यामेलियन सेलबाट निकालिएको, जस्तै हाइब्रिडोमा टेक्नोलोजीबाट निकालिएको छ। अनि म्याम अलिकति स्लाइड ठुलो गराउन मिल्छ? अनि यो स्लाइडमा चाहिँ अनि जेनेरिक नेम्सहरू छ अनि त्यसको कम्पनी कहिले निस्क्यो र के को ट्रिटमेन्टको लागि म्यामेलियन सेलहरू युज गरिएको छ भनेर दिएको छ। यो इक्जाम्पलहरू छ यसमा। यो म्याम यो सबै भन्नुपर्छ?
हुन्छ, भन न, एक दुईवटा भनिदेऊ न, एक्सप्लेन गरिदेऊ।
हाइब्रिडोमा सेल्स कल्चरबाट निस्केको भनेको म्युरोमा। अनि म्याम मैले यो एक्ज्याक्टली के को लागि भनेर राम्रोसँग पढेको छैन है। अनि त्यही यहाँ लेख्न त लेखेको छ तर टोटल रिसर्च चाहिँ मैले गरेको छैन। अनि त्यो म्युरोमा भनेको चाहिँ एक्युट किडनी ग्राफ्ट रिजेक्सन्स इन कार्डियक एन्ड लिभर ट्रान्सप्लान्टको लागि ट्रिटमेन्ट हुँदो रहेछ यसबाट। अनि प्रोड्युस बाई सेल कल्चर जस्तै काइमेरिक एन्टिबोडिजहरू भनेको छ यहाँ इक्जाम्पल हो नि। अनि प्रोड्युस्ड बाई सेल कल्चर रिकम्बिनेन्ट ह्युमनाइज्ड एन्टिबोडी भन्नेहरूको नि छ। डिफ्रेन्ट टाइप्सको डिजिजहरूको ट्रिटमेन्टको लागि युज गरिन्छ। यिनी भ्याक्सिन्सहरू हो, यिनी ड्रग्सहरू हो। म्याम नेक्स्ट स्लाइड। यो चाहिँ नन म्यामेलियन सेल्स जस्तै ब्याक्टेरियाबाट ब्याक्टेरिया नन म्यामेलियन सेल्सहरू भनेको जस्तै हामीले एन्टिबोडिज अनि भ्याक्सिन्सहरू ब्याक्टेरियाबाट अनि प्लान्ट सेल लाइन, इन्सेक्ट सेल लाइन, इस्ट सेल्स लाइनहरूबाट पनि निकाल्ने गरिन्छ। यो इक्जाम्पलहरू चाहिँ ब्याक्टेरियाको प्रोडक्सनबाट ब्याक्टेरियाको सेल लाइनबाट निस्केको प्रोडक्टहरू हो। जस्तै अमालिसबाट अनि प्रोडक्ट अनि ब्याक्टेरिया अनि यसको एप्लिकेसन्सहरू दिएको छ। अमाइलेज थर्मोफिलिक ब्यासिलस स्पेसिसको अमाइलेजबाट निस्केको बाट निकालिएको त्यसको अमाइलेजबाट निकालिएको के अरे प्रोडक्ट हो यो, जुन चाहिँ अमाइलेजलाई ब्रेकडाउन गरेर माल्टोजमा इन्ब्रिभिङ टु ब्रेकडाउन्स अमाइलेजलाई चाहिँ माल्टोजमा ब्रेकडाउन गरेर निकाल्ने गरिन्छ यसको एप्लिकेसन भनेको। अनि जस्तै एनिमल्स प्लान्ट सेलबाट इन्जाइमहरू पनि निकाल्ने गरिन्छ अनि भ्याक्सिन्सहरू पनि निकाल्ने गरिन्छ। प्लान्ट सेलबाट निकालेको के अरे इन्सेक्ट सेलबाट निकालेको भ्याक्सिन्स चाहिँ के अरे क्यान्सर्स भ्याक्सिन्सहरू निकालिएको छ। जस्तै सेरिब्रिकल क्यान्सरको लागि निकालिएको छ इन्सेक्ट सेल्सबाट। अनि इस्ट सेल्सबाट चाहिँ इन्जाइमहरू निकालिएको छ, पेप्टाइड्सहरू निकालिएको छ अनि क्लोटिङ फ्याक्टरहरू पनि निकालिएको छ इस्ट एक्सप्रेसनबाट, इस्टको सेल लाइन्सहरूबाट। म्याम नेक्स्ट स्लाइड। अदर नन म्याम जस्तै इस्ट एक्सप्रेसनबाट स्याक्रोमाइसिस सेरिभेसिसहरू इस्ट हो, जसबाट चाहिँ हाई प्रोटिनहरू सेल ग्रोथको लागि हाई प्रोटिनहरू निकालिएको छ अनि क्लोटिङ फ्याक्टरहरू पनि निकालिएको छ। पेप्टाइड्सहरू एल्बी ग्लुटाइड भन्ने पेप्टाइड निकालिएको छ। इस्ट एक्सप्रेस सिस्टम एचिभ ऱ्यापिड सेल ग्रोथ एन्ड हाई प्रोटिन्स एल्ड विथ स्ट्रेट फरवर्ड प्रोडक्सन scalability and without the need for animal derived growth factor. Each cell's line may also be used to produce protein that cannot be obtained from E. coli due to the problems associated with folding and stereochemistry. The expressions of a fully humanized slighted glycoproteins in glycoengineered yeast consist a major advance in the use of yeast expression system for biopharmaceuticals manufacturing. Plants and insect cell expression systems also able to produce proteins with complex glycosylation patterns. However, the glycans structures product are significant different from those products in humans. इन्सेक्टबाट निकालेको चाहिँ सेर्भिकल क्यान्सरको लागि निकालिएको छ। अनि प्लान्ट सेलबाट निकालेको इन्जाइम भनेको ट्यालिग्युसेरियस अल्फा भन्ने छ, जुन चाहिँ बायोथेरापेटिक्स मेथडबाट निकालिएको प्रोडक्ट हो। एनी क्वेसन?
सोध है, के क्वेसन छ भने सोध है। यो उनीहरूको टपिककै हो, यो टपिक रिलेटेड हो है यो।
सेल लाइनबाट चाहिँ कसरी भनौँ न जुन यो एन्टिथेरापेटिक्स बनाइन्छ नि यो कसरी बनाइन्छ लाइक एक्ज्याक्ट भनौँ न प्रिसाइजली यसको प्रोसेस चाहिँ के हो?
शारदा तिमीले त सबै होइन योबाट यस्तो निकालिन्छ भनेर सबै भन्न त भन्यो होइन, तर एउटाको एउटा फुल प्रोसेस होइन सजिलो कुनै एउटा इन्सेक्टबाट अथवा क्यान्सर सेल लाइनबाट होइन कुनै केही निकालेको होइन कसरी प्रोसेस तिमीले भन्दा राम्रो हुन्थ्यो। तिमीलाई अलि आउँछ भने भनिदेऊ।
म्याम मलाई आउँदैन, मैले हेर्न चाहिँ हेरेको थिएँ तर आउँदैन।
बुझेन होला होइन?
हजुर।
यसो गर न त, अब ऋषभले नै सोधेको छ होइन हाउ भनेर, ऋषभले नै नेक्स्ट टाइम त्यसैको एन्सर खोजेर प्रेजेन्टेसन दिँदा कस्तो हुन्छ?
हुन्छ म्याम।
हुन्छ ऋषभ?
हुन्छ म्याम।
सक्छौ नि होइन?
यस म्याम।
ल कुनै एउटा होइन कुनै एउटा प्रोडक्ट चाहिँ कसरी बन्छ भनेर तिमीले एउटा कुनै राम्रोसँग एक्सप्लेन गर्दा पनि हुन्छ है। सो अरू 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 केही क्वेसन? अरू केही भन्नु छ? छैन है। अब नेक्स्ट थियो शिला सायद प्रेजेन्ट छैन, क्रिस्पर क्यासको बारेमा थियो होइन। ठिकै छ। ओके सो आजको प्रेजेन्टेसन चाहिँ शारदाको र रुबीको है। केही क्वेसन सोध्नु छ भने भनिदेऊ। अनि विकासलाई पनि मैले कुनै प्रेजेन्टेसन गर्न दिएको थिएँ जस्तो लाग्छ है। त कहिलेकाहीँ मात्र आउँछ, क्लास रेगुलर आउँदैन।
सेनिटाइजेसन टेक्निकको
रेडी छ? तिमीसँग प्रेजेन्टेसन रेडी छ?
प्रेजेन्टेसन रेडी छ म्याम। मेरो ल्यापटपबाट माइक प्रब्लम हुन्छ, मैले हजुरलाई टेक्स्ट के त्यो मेल गरिदिन्छु अनि हजुरले त्यो सेयर गरिदिनु।
स्योर ल पठाऊ है त, मेल पठाऊ ल।
Ma'am, आयो?
छैन विकास।
मैले म्यामलाई मेल गरेको थिएँ।
साउन्डै आएको छैन कि
रेडी होइन आयो होइन
आयो म्याम
Hello, am I audible?
Yes, you are.
Okay, good morning, respected ma'am and my fellow friends. Today I am going to present some views and some reports on the topic sterilization technique. First of all, ma'am, slide please, slide change. First of all, sterilization refers to that process which removes, kills, or deactivates all forms of life. As we are the student of microbiology, we are quite familiar with this term sterilization, and it means that any process that removes, kills, or deactivates all forms of life. And what's the main aim of sterilization? That we have to know. The main aim of sterilization is the reduction of initially present microorganisms or other potential forms of pathogens. Pathogens are those microorganisms which can cause disease. And sterilization is quite distinct from disinfection, sanitization, and pasteurization. Here, the term disinfection means to remove the infection and sanitization means to inhibit the microorganization, and pasteurization means completely kill the microbes present in the liquid form by evaporation and followed by cooling process. In these methods, we generally reduce rather than eliminate the forms of microorganisms or biological agents present in the substance or any medium. Uh, After sterilization, an object is said to be sterile or aseptic. We are quite familiar with these terms sterile or aseptic, and we are using the equipments that are used by the health professionals. All of them are sterile or aseptic. Beside that, we prefer to use sterile objects during our lab projects. And slide since and we have there's a long history of this process. In the 460 in the 460 BC, Hippocrates was born, and he was the first to separate the medicine from philosophy, and he had disproved the idea that disease was punishment of the sin. Before the time of Hippocrates, there was belief that any disease was punishment of the sin. He disproved that philosophy, and he also advocated the irrigation of wounds with wine or boiled water. That is being practiced in the today's day too. We are using 70% ethanol, that is alcohol, to clean or sanitize our hand, which was which was pioneered by the Hippocrates uh, in the early BCs. And the Image Samuels was Hungarian obstetrician who advocated the value of hand washing and fingernail scrubbing in 1847. And moving to 1862, French chemist and microbiologist Louis Pasteur discovered the technique of pasteurization, in which he published his findings on how germs can cause disease, and which was later used as pasteurization technique. And in 1867, an English physician, Joseph Lister, reduced the mortality rate of his patients by using carbolic solution spray as he operated, and then he used those carbolic solution in the wound so as to recover the patients, and in this way he reduced the mortality rate of his patients. And slide, please. Uh, Louis Pasteur's pupil and his collaborator developed the first pressure steam sterilizer, or what we call autoclave in modern days, in 1876. This is the milestone that the sterilization technique had achieved. And later on, the research of Robert Koch and his associates on disinfecting properties of steam and hot air marked the beginning of the science of disinfection and sterilization in 1881. And this proved the importance of today's modern day sterilization technique by using the steam or hot air. And Esiclap created the first rigid instrument container, originally made up of stainless steel in Germany in the early 1900s, responding to the needs of the military hospitals and aid stations. He manufactured chrome-plated containers for safe transport of sterile materials, what we call autoclave in the today's day. Next slide, please. Basically, there are three methods of sterilization. They are the physical method, chemical method, and the filtration. And the slide, physical methods of sterilization are heat sterilization, filtration, and other the form in the form of the waves. They are found sound waves, pressure, or sunlight. In the heat sterilization, there are two types of sterilization: moist heat sterilization, and it is the most effective and widely used method for sterilization. And it is done at the temperature below 100 degrees centigrade, at a temperature of 100 degrees centigrade, and at temperature above 100 degree centigrade. They are further classified as moist air sterilization and dry heat sterilization too. And for the filtration, we use two types of materials or medium. They are liquids or gas. Filter sterilization is done of liquids as well as gas, and irradiation by the use of the radiation. They are ultraviolet radiation or ionizing radiation. Slide change. Talking about heat sterilization, it is the most effective and widely used method for sterilization process, where the bactericidal activity results through the destruction of enzymes or and other essential cell constituents. Here, bactericidal activity refers to the killing or inhibiting the bacterial activity. The term bactericidal refers to that. And the effects of heat sterilization generally occurs more rapidly in a fully hydrated state, as it requires a lower heat input with low temperature and less time under high humidity conditions where the denaturation and hydrolysis reactions are predominant, rather than in a dry state where oxidative changes takes place. Generally, we prefer to do this technique at a low heat, at low temperature, and within less time, because under high humidity condition, along with under high humidity conditions where the denaturation and hydrolysis reactions occur more frequently, more effectively than in the dry state, where in the dry state where oxidative changes only takes place. Under circumstances where thermal degradation of a product is possible, it can usually be minimized by adopting a higher temperature range, as the shorter exposure time generally result in a lower partial degradation. In this method of sterilization this method is applicable to the thermostable product. Still, it can be applied to both moisture sensitive and moisture resistant product, for which we can use dry heat sterilization up to 160 to 180 degrees centigrade, and for moist technique we generally use temperature ranging from 121 degrees centigrade to 134 degree centigrade, where we use heat sterilization technique. Ma'am, slide. For moist is talking about the moist heat sterilization, it is one of the most effective methods of sterilization, where the steam under pressure acts as an bactericidal agent. Here, the steam under pressure acts as bactericidal agent. The temperature range of this method is 121 degrees centigrade to 134 degrees centigrade under high pressure. High pressure also facilitates the rapid penetration of heat into deeper parts of the material, and moisture present in the steam causes the coagulation of protein, causing the irreversible loss of function and activity of microbes, so that bactericidal activity can be increased and we can inhibit the effect of microorganism. The high temperature short time cycles not only often result in lower fractional degradation, but they also provide the advantage of achieving higher levels of sterility assurance due to more significant inactivation factors. We generally prefer this method, or we consider this method as the higher level of sterility assurance. We can assure this method as high level for eradicating the bactericidal agents. And slide. And it is the most commonly used standard temperature cycles for clinical porous specimens such as surgical dressings and bottle fluids are 134 degrees centigrade for 3 minutes and 121 degrees centigrade for 15 minutes respectively. Autoclave works on the principle of this technique, that is moist heat sterilization technique through the generation of steam under this pressure. In this method, the microorganisms are killed by coagulating their proteins, and this method is much more effective than dry heat sterilization, where microbes are killed through oxidation. And this method is generally used in the pharmaceutical and medical sectors, as it is used for the sterilization of dressings, seeds, surgicals, and diagnostic equipment, containers, and aqueous injection of ophthalmic preparations and irrigation fluids, in addition to the processing of soiled and contaminated items, so that the patient could not bear the risks of infection by microorganisms. So moist heat can be used in the sterilization at different temperatures. At the temperature below 100 degrees centigrade, the sterilization technique employed at 100 temperature below 100 degrees centigrade involves pasteurization. In this process, all non-spore-forming microbes are killed in milk by subjecting the milk to a temperature of 63 degrees centigrade for 30 minutes or 73 degrees centigrade for 20 seconds, the flash method. In pasteurization, however, all the pathogenic organisms are killed. The principle of pasteurization is the logarithmic reduction in the number of viable microbes so that they can no longer cause any disease. Serum and body fluids with congealable proteins are also sterilized at 56 degrees centigrade for 1 hour in water bath. And it is the most commonly used standard temperature time cycles for clinical porous specimens too. And could you please change this slide? And and we are talking about dry heat sterilization, and it is the process of removing microorganisms by applying moisture-free heat, which is appropriate for moisture-sensitive substances. The substances which are moisture sensitive, they can be sterilized by this process, and this process is based on the principle of conduction, that is the heat is absorbed by the outer surface of an item and then passed onward to the next layer, and ultimately the entire item reaches the proper temperature which is required to achieve sterilization, and which is capable of killing the microorganisms present on the surface of the equipment. And dry heat, dry moisture less, it destroys microorganisms by causing a denaturation of proteins and also lyses the proteins in many organisms, causing oxidative free radical damage and causing dying of cell, and can even burn them to ashes as in sterilization. It is used for the sterilization of materials so which are difficult to sterilize by moist heat sterilization for several of the reasons. Substances like oils, powder, and related products cannot be sterilized by moist heat because moisture cannot penetrate into deeper part of the oily materials, and powders are destroyed by moisture. So we prefer to use dry heat sterilization for those substances rather than moist heat sterilization. Next slide. And then we'll move to radiation sterilization. It has two types. Uh, they are non-ionizing and ionizing. Non-ionizing generally refers to hot sterilization technique. It has also two types. They are infrared rays sterilization. This technique is used for rapid mass sterilization for the pre-packed items such as syringes, catheters, and UV rays is generally used for the disinfecting enclosed areas such as entryways, operation theaters, and lab. And generally, we do this disinfection or sanitization method in our laminar hood via UV rays too. Like, and the second is ionizing or cold sterilization. One slide, please. And this is done generally done by the gamma rays and using gamma rays and X-rays. They are used for sterilizing plastic, syringe, swabs, animal feeds, oils, greases, fabric, and metal foils. The main advantage of radiation sterilization is that it is a clean and dry process, and it ensures full exposure of objects from all directions, and we can disinfect or we can sanitize the object properly. And the major disadvantage of this technique is that it poses threat to human. It can The long exposure of these rays can cause cancer to the human, and this process is quite lengthy as compared to the other methods of sterilization. And for this technique, for and to run this technique, we require very qualified person in order to achieve proper assurance of sanitization, as well as not to get uh, not to have some effects from the exposure of these rays, so that we require very qualified person. And we have clinical methods for sterilization. There are gaseous sterilization and liquid sterilization. In the gaseous sterilization, we use ethylene oxide and formaldehyde gases. For liquid sterilization, we use alcohol and phenol. Talking about the gaseous sterilization, we use ethylene oxide as well as formaldehyde. Ethylene oxide is mainly used to sterilize medical and pharmaceutical products that cannot support conventional high temperature steam sterilization, such as the devices that incorporate electronic components, plastic packaging, or plastic containers. Generally, we prefer this technique for the plasticware. And this method uses an automatic device filled with ethylene oxide gas at temperature below 100 degrees centigrade to sterilize complex and delicate material. And ethylene oxide also destroys microorganisms by chemically reacting with their nucleic acid. And the slide 16. And the major advantage of this technique is that we use fully automated device, so that we can assure high efficiency and 100% result. And the disadvantage of this technique are this process is quite complex and time consuming, and if we do some negligence during the process, we have to we might get infected from carcinogenic effects, or we might uh, get some cancer causing agents to our body. And the second method for this technique is formaldehyde gas. It is also another low temperature method for sterilization of heat sensitive items like the plastic wares. Uh, it is organic compound which is a byproduct of the metabolism of many organisms, and is commonly found in fresh air, in water, foods, industrial products, and fabrics. This method is considered even more dangerous than ethylene oxide, and is and so it is less commonly used for sterilization technique. And this sterilization is used where sterilization by steam or high temperature is not possible. It is soluble in water, and its inactivation power is greatly improved by the presence of humidity. It is most commonly used as disinfectant, but sometimes formaldehyde is used as sterilizing agent. The process known for this technique is low temperature steam and formaldehyde (LTSF). In countries such as United Kingdom, Germany, Sweden, Denmark, and Norway, sterilization by LSTF is accepted but not common. On the other hand, in several countries, formaldehyde as a sterilizing agent is discouraged due to its higher carcinogenic property. LDSF has not been approved by FDA for use in healthcare facilities in the United States of America. Slide. The major advantages of this technique is it is very reactive molecule, and its time cycle is quite faster than the ethylene oxide, and it is quite a cheaper technique as compared to the ETO. After sterilization, most loads are available for immediate use, and the disadvantages are the vapor that arise from the formaldehyde is quite irritating to the eyes and has weak penetrating power compared to the ethylene oxide, and it operates on higher temperature rather than ethylene oxide. Its residue can remain on the sterilized goods if the rinsing phase is not 100% efficient. This can be harmful to the patients. Slide. And the second one is liquid sterilization. I think all of us are familiar with this technique by using alcohol. Alcohols are quite effective disinfectant for many reasons. They evaporate quickly without leaving any residues on the surface. They are capable of dissolving lipids, which make them effective against lipid-wrapped viruses such as HIV, SARS-CoV, and hepatitis A. They are inexpensive and relatively easy to handle, although their vapors are flammable. Ethanol and isopropyl alcohol, both members of the alcohol family, have similar disinfectant properties, and they can ethanol is the type of alcohol present in alcoholic beverage, and isopropyl alcohol is also known as isopropanol, 2-propanol, or rubbing alcohol. When used as disinfectants, both are typically at the concentration of 70% in water, and what we call them is sanitizer. Slide in. Phenol. Phenol is one of the oldest antiseptic agent. It is aromatic alcohol, and they acts by damaging cell membrane, thus releasing the cell contents and causing lysis. Phenol is commonly found in mouthwashes, scrub soaps, and surface disinfectants. Phenols are used for decontamination of the hospital environment, including laboratory surface and non-critical medical items. The smell of phenol, what we love to call, is the hospital smell, and it smells like the hospital. Uh, so it is generally used to disinfect or sanitize the hospital, and its smell is preferred to call as hospital smell. Lighting. And the third method for the sterilization technique is the filtration. It is unique among sterilization technique that it removes rather than destroys the microorganism. Also, it is also capable of preventing the passage of both viable and non-viable particles, and thus can be used for the both clarification and sterilization of liquid as well as gases. The primary mechanism involved in this filtration are sieving, adsorption, and trapping with the matrix of the filter material. Filtration uses membranous filters that have tiny pores, and those tiny pores let the fluid pass through, but prevent bigger particles such as bacteria from passing through the filter. Therefore, the smaller the pore, the more likely the more likely the filter is to stop more things from going through it. Certain types of filter also have an essential role in sterility testing, where they can be employed to trap and concentrate the contaminating organism from the solutions under the test. These filters are then placed in a liquid nutrient medium and incubated to encourage growth uh, and turbidity of the microorganism. Uh, I had used I had cited all these informations from www.study.com and www.researchgate.com and WHO Glossary. Thank you. I'll be more than happy if you have any queries.
Any queries? छैन जस्तो छ है। ल विकासको पनि प्रेजेन्टेसन स्टेरिलाइजेसनको सक्यो। सबैले बुझ्यो होला हैन टेक्निक्सहरुमा के पर्छ भनेर। हिट, मोइस्ट हैन, स्टेरिलाइजेसन, त्यस्तै गरेर केमिकलमा फिल्ट्रेसन हैन, यिनीहरु सबै टेक्निक्सहरु ठिक छ सबैले बुझ्या छैन ओके ल अब नेक्स्ट कसको पाली हो त प्रेजेन्टेसन दिनलाई उत्सव रेडी छौ
हजुर म्याम
तिमी रेडी छौ प्रेजेन्टेसन दिनलाई
होइन म्याम मैले
होइन कि अर्को पालीमा तिमीलाई म टपिक
दिन्छु म्याम अर्को पालि भन्नु त टपिक
लाइक अब के टपिक दिउँ भन्दाखेरि होइन अहिलेको करेन्ट सिचुएसनकै बारेमै राम्रो हुन्छ होइन सो यस्तो गरौँ न त लाइक यस्तो छ होइन अब डिफ्रेन्ट एराहरूमा चाहिँ डिफ्रेन्ट पेन्डेमिकहरू आइरहेको थियो होइन स्पेनिस फ्लू अनि अर्को ब्ल्याक डेथ होइन अनि अहिलेको कोभिड होइन यो तिनटैमा तिमीले यसो देऊ न त कस्तो थियो डेथहरू होइन डेथदेखि लिएर त्यसको सिम्टम्सहरू कस्तो थियो केले कज भएको थियो यो सबैको बारेमा तिमीले चाहिँ हल्का फुल्का एउटा स्लाइड बनाएर प्रेजेन्टेसन देऊ न त है अनि के गरेर त्यो चाहिँ एलिमिनेट भयो कि यो कसरी डिसएपियर भयो होइन अब कोभिड-19 पनि पहिलाको जस्तै स्पेनिस फ्लू जस्तै गरेर डिसएपियर हुनसक्छ कि होइन त्यो चान्सेसहरू छ कि के छ भनेर यसो इन्फर्मेसनहरू लिएर आएर तिमीले प्रेजेन्टेसन देऊ है त एक्ज्याक्ट टपिक नै त भएन तर तिमीले चाहिँ यो तिनवटा यो छ नि यो तिनवटा
महामारी भनेपछि अहिलेसम्म आएको महामारीको यसमा कसरी आयो कसरी गयो भनेर
है कतिको चान्सेस छ त यो कोभिड हराउने चान्सेस छ कि यो सधैँ नै रहिरहने हो कि होइन यस्तो अब अलिकति डिफ्रेन्ट केही टपिक लिएर हो है त अनि तिमीले चाहिँ त्यसमा इभन त्यो कोभिडको भेरिएन्टहरूको बारेमा पनि एक्सप्लेन गर्न सक्छौ के रहेछ त यो भेरिएन्ट भनेको के रहेछ भनेर एक्सप्लेन गरिदेऊ है त ल
हुन्छ म्याम हुन्छ कहिलेसम्म म्याम
अब हाम्रो जेस कोर्स सकिसकेको छ होइन कोर्स सकिसकेको छ अब तिमी नेक्स्ट क्लासमा अब आज फ्राइडे ट्युजडेको क्लासमा है त ल आउनु नि फेरि है अच्छा नहुनु नि
लाइन गएन भने नेट गएन भने आउँछु हुन्छ
ल ल ल हुन्छ ल आजको क्लास यही नै भयो होइन एटेन्डेन्स लिन्छु अनि बिदा गरौँला